everybody for coming today. So I will talk about my work on hydrogel-based soft robot that swims by minimal actuation by harnessing flutter instability. So let me introduce some motivation. Even very simple organisms, such as single cells, have the ability to swim by quite complex shape change, and they can propel themselves in viscous environments by just changing shapes. And also recalling the constraints of low-Reynolds number hydrodynamics, these shape changes must be non-reciprocal in order to allow for motility, meaning that the motion must be non-symmetric by time reversal. And for example, here we see a sperm cell which swims moving its flagellum, and there are flagellated cells, organelles, which have a different flagellum. And indeed, these elongated filamentous structures are ubiquitous in nature, in living organisms, either as single flagella or in ciliary carpets like this, where we can see many filaments that move in this metachronal, forming these metachronal waves on the outer surface of a paramecium, which is a big cell, which again swims, moving all this cilia. In living organisms, this motility is possible thanks to the conversion of chemical energy into mechanical work by specialized and very complicated molecular motors, which are topic in itself. But in the last decades, we have seen the rise of active materials, which are artificial materials that can be activated by an external stimulus and can perform some form of motion, shape change. For example, here is a liquid crystal elastomer which, under illumination, starts to bend and snaps. In the video, or a work in our group of some years ago. These are hydrogels which respond to chemical gradient and again change their shape. And taking the wrong button every time. It is quite natural to ask if these materials can be employed to replicate the motion of cells in biomimetic robots. The first, this one, is a seminal work where, just to get the context, that is a red blood cell where they have attached a filament made of magnetic particles, which are attached to one another with DNA filaments. Then there is a magnetic field that keeps the filament straight and a transverse magnetic field that oscillates it, and this micro robot swims in this way. Other strategies for this kind of work is this is a hydrogel, piece of hydrogel, like the material shown here, which is in a container that is shaken, and thanks to its geometry, the tail oscillates and it propels itself in the fluid. And also active materials employed in the biomimetic replicas of this kind of structures. So ciliary arrays made of, this is again a hydrogel with magnetic particles, and then there is a magnetic field that is rotated below the system, and so the cells perform this motion and move the fluid in a direction. So in all these examples, which are fascinating, there is the need to actuate these active materials in a time-dependent way in order to obtain these time-dependent responses. Which makes sense. But more recently, some strategies have been employed to obtain time-dependent response from no or constant stimuli. For example, here is a hydrogel where this is filled with the chemical solutions which undergo a cyclic chemical reaction, so the pH changes periodically, and so the hydrogen responds periodically. So with no stimulus, external stimulus that is, this can change shape periodically and walk. Another work is this, again an hydrogen which is actuated by a laser like this, and the laser is constantly pointed where the band is, and this tail flaps up and down, and so the laser hits below and up, and so there is automatic oxidation that occurs. Then these are not active materials actually. These are two passive pieces, a passive dielectric particle and two passive elastic tails. But here is another strategy. This is activated by an electric field, which is constant, very high electric field. And there is an instability, which is known since the 1850s or so, which induces a rotation in the dielectric particle in presence of some of their unconditioned, let's say. So by attaching these filaments, they can make this swing. And finally, an example again from our group. This is again the same hydrogen, almost the same hydrogen as before. This is activated by a constant electric field, which points up. And again, thanks to an instability, this self-oscillatory behavior emerges. So in this work, we propose to along these findings, we propose a new mechanism to build a swimming hydrogel robots where ondulatory swimming is achieved by harnessing flutter instability, again under the constant electric field. This is done both by mathematical models and also by proof of concept experiments. And of course, the two aspects develop and influence each other all the way, but I will present experimental findings first, and then model, which tries to explain and predict the experiments. So, as I was saying, the material that we are using is an hydrogel, which is essentially it's a polymer, which is like a sponge filled with water. The base polymer is made by acrylamide monomers, which cross-link with this acrylamide. And then some monomers of this other substance, which essentially introduces fixed negative charges in the polymeric network, which gives the material its active nature. The polymer does not create by itself. The reaction is initiated by a photoinitiator, which is essentially a chemical molecule which, under illumination, breaks down, releases some very reactive free radicals, which allow to overcome the energy barrier required to polymerize the molecule. And in order to be able to create different geometries for these objects, we employ a photolithographic technique, which means that we have a UV projector, which is a projector like that, which projects UV light. So we can draw the mask of the shape that we want to obtain. This is projected in a microscope, which focuses this light in the solution of monomers, and where the light hits, the free radicals are released, and so the reaction starts, and we have localized polymerization of the material. These are pictures of the setup. So this is the microscope, and the spot of light is where the sample is being prepared. Okay, after the samples are prepared, the experiment is performed in an electrolytic cell, because we need to activate this material by an electric field. So we have two electrodes, to which we apply a electric potential, and an electric field is generated between them in a solution of salty water. So the ribbon of hydrogel is placed there, and given the mechanism of what happens is not completely understood, but under the electric field, there is migration. A plausible explanation is that there is migration of the mobile ions in the cross section of the ribbon. So we have a differential swelling, which causes the development of curvature in the region. Then we can, so at this point we can perform the experiment, but we need to see what's going on, because these are hydrogels, so these are made of more than 99% water, so they're almost invisible in water. And to see them, we introduce in this network some particles which are fluorescent under the right light. So we illuminate them the right way, and they emit green light. So filtering out the rest of the light, we see only the sun. Okay. So first we talk about the characterization of this material. So the experimental setup now is rotated with respect to the previous drawing, but the filament is there, the ribbon is there, it's aligned so that the thinner side faces the camera. And introducing the electric field, it bends around the soft axis, and it bends toward the cathode, which means the side where the negative side of negative charges are. If we increase the potential difference, we see that the curvature becomes bigger, and it's always localized where the field line is perpendicular to the temple. Now let's see how these equilibrium configurations are reached in time, and how they depend on the initial configuration. So we have a filament that is aligned with the field, and then some are a bit more rotated. We see that by increasing again, the curvature increases, and most importantly, that they all reach their respective equilibrium configuration in the same time. So there is a characteristic time for this phenomenon. And yeah, and then again, the distribution of curvature, the curvature is localized to the filament is orthogonal to the electric field. These observations will allow us to model the behavior of this material later. But okay, now let's see what happens increasing the electric field. We've seen before that a filament ribbon aligned with the electric field for lower values of the potential difference remained straight, while for a sufficiently high potential difference, these small amplitude oscillations emerge, which then develop into higher amplitude oscillations, which stabilize. And the ribbon swims towards the cathode of the electrolytic cell. So this was the initial transient. And now let's try to see what happens when the oscillations are steady. And we see that there is, these are quite regular in period and amplitude and shape and so on. And, well, again, recalling that we are at low Reynolds number, this shape change must be not reciprocal. Indeed, a way to visualize it better is by a chemograph, which essentially means projecting the time variable in one of the two directions here. So, well, the video I think explains it. So we have these two lines, and on this side we see the material points which cross this line in time in that direction, while time is downwards here, and we see the material particles crossing the horizontal line. And looking at this picture close enough, we can see that the shape change of this object is characterized by a backward traveling wave, and that this wave is increasing in amplitude while traveling towards the tail of the field. Okay, so what we set to do is done, but we repeated the experiment many times to collect data about this behavior. So we performed again the same experiment many times. So we collected information about what was the average advancement for each cycle of a shape change and the average period of the shape change. And also we performed experiments with different values of the potential difference, at least in the range that was feasible in our experimental setup. Okay. What we see is that compared to other swimmers at this scale, is quite efficient at swimming. It has an average velocity of half a body length per second, which is quite fast compared to previous attempts. Now we would like to understand what's going on, and for this reason we develop a simple mathematical model which allows us to describe in a more formal way what's going on. So since these ribbons, the motion of these ribbons was essentially limited to plane, the model is for inextensible filaments in plane motion. Its shape is completely described by the inclination of the filament with the horizontal direction. And so its tangent vector can be written in this way, and its position is determined up to a translation. Now we impose balance of linear and angular momentum. The only external force comes from the viscous fluid that is around the filament, and the viscous interaction, it would be quite complicated to solve exactly, because we would need to solve Alter-Stokes problem. So it's in the outer domain, but we can represent, there are reduced theories for viscous drag, which allow to represent the drag as proportional to the velocity of each point of the filament by two coefficients, which have recently been computed for ribbon geometries. So that's what we adopt. As for the bending moment in the angular momentum balance, we adopt a morphoelastic rod theory. So bending moment is proportional to the difference between this, the current curvature, theta prime, and a spontaneous curvature, which is the internal degree of freedom that encodes the activity of the material. This, okay, for this we need to, again, constitutively prescribe an evolution law, and in the spirit of having a simple model, we use a simple evolution law. This hides the true complexity of the hydrogel and the chemistry and the physics that happens there. But nonetheless, it represents the main features that we observe in the characterization of the material, namely that we have a target curvature, kappa A, which is higher for higher electric fields. There is a characteristic response time, which is denoted tau, and by modulating the target curvature by this sine theta, we represent the fact that the curvature localizes only where the filament is orthogonal to the field. Finally, we close this system with boundary conditions, which represent the fact that the filament is free both sides. We can cast these components, and by running finite element simulations, we can see that for some choices of the parameters, nothing happens, and for other choices, something happens, the filament swings. But we would like to compare these results with the physical experiments, and for this we need to appropriately choose the parameters. The geometric measures of the ribbons are measured, while there are three parameters that are very difficult or impossible to measure from experiment. So to determine them, we fit the numerical results with the experimental measurements by minimizing this cost function, which accounts for the discrepancy in net advancement per cycle, which is what we measure here, average period, and lateral displacement of the tip, so how much the tip goes up and down. This is done, minimization of this cost function. Evaluation of this cost function means running a finite element simulation, extracting the data, comparing the experiments, and we minimize it by a gradient-based method, leading to the determination of the remaining three parameters and the value of the cost function 3 minus 2, which means that in these three quantities, the discrepancy is about 15% between model and experiment. That, considering how simple the model actually is, was quite satisfactory. So let's see the simulations which should correspond to our ideal experiment. we see that the tail starts to oscillate, then small amplitude oscillations develop into our limit cycle. Okay, and now we can compare also the shape change with the same chemographs as before, and we see that they agree quite well. Of course, the comparison between the two was, I mean, the fitting was done only on the motion of the tip, while the whole motion is captured quite well. Just a small note. I noted that the swimming always occurred towards the cathode. So one thing we thought was that if we rotate the electric field, we could rotate the swimming direction. This is a very long video, so that's around thing. But okay, so here is happening the same thing as the previous simulation. But then we start to rotate the electric field at some point. And the swimmer follows the direction of the electric field. And so by rotating the electric field with constant angular velocity, we can make it go around zero. Now, again, we've seen that the model captures the behavior of what we have seen in the experiments, but we would like to understand why this transition from stationary and straight filament transition to oscillators, to self-sustained oscillations of the proportion. And as to this end, we study the stability of the straight configuration, varying the model parameters. To make things simpler, reduce the number of parameters to identify some characteristic scales for the physical quantities. And by introducing these three-dimensionless parameters, we can recast the governing equations in that form, which only depends on 3 instead of 4 parameters. And okay, then we identify again the trivial equilibrium solution and perturb it with a small amplitude perturbation. And by retaining only first order terms in this perturbation parameters, we leads to coupled linear LPDs. Now the system is at linear order is only governed by 2 of the 3 parameters. And well, we also linearize the boundary condition. Then we look for solutions in this form, where we separate space and time variables, and this omega is the eigenvalue in characteristic frequency with which the oscillates. And again, we can simplify these equations into a fourth order linear ODE. This can be integrated analytically with Mathematica. And imposing the boundary conditions, we get a characteristic equation, which, if one was able to solve it, would give the eigenvalue for corresponding to the values of the system of the 2 model parameters. This equation cannot be solved in closed form, but we can solve it numerically. We need to fix the 2 parameters, and for this choice of parameters, which corresponded to an oscillatory behavior in the finite element simulation, we indeed see that or eigenvalue has a positive real part and non-zero imaginary parts, meaning that any perturbation, any small perturbation will lead to an oscillatory mode of increasing amplitude with these eigenfunctions, which are quite similar to the actual shape of the filament. And well, for these parameters, we also find other eigenvalues. So one is positive and the remaining are negative. Okay. And then repeating this procedure and inspecting the eigenvalues for every combination of the parameters eta and t, we can build this stability map, which tells the behavior of the system for all combinations of the parameters in this range. So, in this work, we have showed that oscillatory swimming, directed oscillatory swimming, can be achieved in a sort of hydrogel swimmer by a minimal actuation, and proved it experimentally with very good performance. And we show that a very simple model can represent and predict the main features of this phenomenon, and also allowed to explain and characterize the behavior observed in experiments as an instance of flutter instability. Quite weak notion of controllability of the system was proven by means of numerical simulations, but we should investigate that better. And in the future, of course, an interesting topic would be to extend this to filaments of plane motion in 3D motion, by studying how the cross-the geometry influences the swimming performance, and also how many of these objects, being cilia or swimmers, interact with each other. And in everything I said, there is a very big abstraction of the actual material, the hydrogen, which is not completely characterized. And again, so the evolution law is mostly phenomenological, but it would be interesting to derive it from the fundamental dynamics of the chemistry and physics of the hydrogen. Thanks for your attention.
Thank you for the beautiful talk and the wonderful images. Are there any questions from here or from home?
Yes. I was just curious, you always talked about the electric field, but you described this experiment as happening into a cell with salt water. So is there actual electric current flowing through the sample?
Through the sample, yeah, because because it's salty water.
Yeah.
So there is a current, there is an electric field and current.
And you expect this phenomenon to be based on the current or to be based on the external electric field?
Good question. I mean, as I said, let's see if I find it. The one model of what's happening is that something causes the mobile ions to migrate in the cross section of the ribbon, and this causes a difference in volume of the two sides of the cross section, which causes it to bend. And that's how the internal moment is generated. Then there are different explanations of this. There are explanations where they say that there is a current which transports ions in the cell, which attach one side and not to the other. Other explanations say that just the field causes migration inside the gel, so there is almost no exchange with them. So the answer is, yeah, I don't know. And
nobody knows.
Probably nobody knows.
Thank you.
Thank you for the very nice presentation. I have just a curiosity about the flight element part. So in flight element simulation, how many special degrees of freedom do you have?
30.
Ah, okay, so
it's not very
it's
intensive.
the also.
Yeah.
I was wondering if some users.
No, because again, the model is as simple as it can be in order to
maybe
represent.
three-dimensional extension.
Yeah, maybe yes. But at this point also, I mean, hydrodynamics is solved, not solved because we just need to compute the velocity, so not much computation.
Io volevo dire che commenta a bit more on the extension from 3D. You already have some ideas what would you need to change to be able to control it in 3D.
Yeah, I mean there is to just go 3D should not be difficult. I'm doing it now because it's again just three-dimensional theory, so these equations are a bit more complicated. You have to like keep track of more angles, but the only real challenge I think would be to extend this evolution law, which again is phenomenological. So in 2D, you see from experiments what is going on. Yeah, I think the part where I'm not really sure is how to extend this to 3D in a consistent and mostly representative way. But as for the model, the rest of the model, no.
And the electric field would mean you need to control this from 3D.
Ah, well,
Simply using the forward direction of the electric field.
that's the idea, but yeah, it should be actually again, as I mentioned, this is an aspect which could be studied further because there is a slight delay between the reorientation of the electric field and the reorientation of the swimmer because it takes some cycles in order to reconfigure the way, so there is a delay. And also there is like an angular velocity of the electric field out of which it cannot follow the electric field. Strange things happen. But maybe it would be interesting to characterize these things.
If there are any questions from here or from home, I would thank Ariel for the wonderful talk and thank you.